Dear students, in the previous lecture we have studied about the transport of water. Now, let us study about plant-water relations. As you all know that water is essential for plants. Before we go into the different type of relations that are found in the plant, let us define certain terms. The first term that we are going to define is water potential. It is the difference between the free energy of water molecules in pure water and the energy of water in any other system. That means that it is the potential that the water contains in a pure solution or in a plant cell or tissue. The second term that we are going to define is solute potential. It is defined as the amount by which water potential is reduced as a result of presence of solute. Another important feature of solute potential is it is always negative, and at atmospheric pressure the water potential is equal to solute potential. The third potential is the pressure potential. It is hydrostatic pressure developed in vacuole when plant cell wall exert a pressure on cellular contents. There are other terms that are important in plant-water relations. They are defined as the actual pressure exerted by protoplasm against the cell wall. The second, the wall pressure. The cell and elastic. It exerts an equal and opposite pressure against the expanding protoplasm. Now, let us see the osmotic relations in three physical states of cells according to water potential. Let us first see in the case of turgid cell that the net movement of water into cell is stopped in the case of turgid cell, and the water potential is equal to osmotic potential plus pressure potential. That is equal to minus 10 bars plus 10 bars, that equals to 0 bars. But in the case of flaccid cell, turgor pressure is 0. Now, let us see with the help of this equation. Water potential, it is equal to osmotic potential. So, the net pressure is 10 bars. Now, let us see the osmotic relations with the help of the following diagram. In the diagram that shows the osmotic potential, turgor potential, water potential, and how the movement of the molecules take place. Now, let us learn about osmosis. Let us first define the process. It is the diffusion of water molecules from dilute solution to concentrated solution when the two are separated by semi-permeable membrane. This we can demonstrate it by a simple diagram as follows. Here we see the two systems, one having the water molecules, the pure solvent, and the other having a sucrose solution. And the two systems are separated by differentially permeable membrane. There is a net movement of water molecule from pure solvent to sucrose solution. Now, next we can again have an osmosis demonstration in the following diagram as follows. Here you can see distilled water, and there is a 3% solute solution which is having a selectively permeable membrane. When this is put into distilled water, the solution stops rising when the weight of the column equals osmotic pressure, and here is the rise of the salt solution. So, there is movement of water molecules from a region of high concentration to a region of low concentration. So, if we summarize the osmosis process, we can see that the movement of water molecules is from the region of pure solvent water to solution, from dilute solution to concentrated solution, high free energy of water to low free energy, higher water potential to lower water potential. Now let us learn about the types of osmosis. The osmosis is of two types, endosmosis, exosmosis. Let us see in the following diagram about the two processes. In the first diagram, we see the turgid cell. Water enters by osmosis, vacuole swells up and pushes against cell wall. In second diagram, you see the flaccid cell. What does these arrows indicate? Yes, the water lost from cell. Vacuole shrinks, cell loses shape. Now, have you heard about the term osmotic pressure? Yes, it is equivalent to pressure exerted upon solution to prevent the flow of solvent into it across a semi-permeable membrane. It is expressed as bars with positive sign. Osmotic pressure of water, pure water, is 0. Now, let us see the importance of osmosis in plants. So, it helps into cell to cell movement of water, in the absorption of water, in the maintenance of shape of the plant cells, turgidity of roots in penetration into soil, resistant of plants to drought and frost, opening and closing of stomata. Now, let us see the behavior of plant cells in the surrounding solution. There are three types of solutions: hypotonic solutions, isotonic solutions and hypertonic solutions. Now, in hypotonic solution, the external solution is more dilute than cytoplasm. In isotonic solution, the external solution balances osmotic pressure of cytoplasm, and in hypertonic solution, external solution is more concentrated than surrounding solution. Now, let us see the three types of solution with the help of a diagram. In the hypertonic solution, the water moves out, so we call it a plasmolysed solution. In the other case, the water moves in, that is the flaccid cell, and here the water when moves in, it becomes the turgid cells. A very important process that we see in the plant is plasmolysis. Now, this plasmolysis is the shrinking of cytoplasm in the plant cells. This can be explained in the following diagram. The diagram shows complete cell when the plasmolysis has not taken place. When it is put into the hypertonic solution, the cytoplasm shrinks. A very important process that found in the plant cell is plasmolysis. Plasmolysis is the shrinking of cytoplasm within the plant cell. Now, let us see the process of plasmolysis with the help of the following diagram. In the diagram, you see a turgid cell and then a plasmolysed cells, and then the plasmolysis complete, that is the shrinking of cytoplasm. Now, what is the importance of plasmolysis in plant? Now, this plasmolysis is used in the preservation of pickles and jams. It is also used to prevent growth of plants in the cracks of walls by spraying salt. The last process that we are going to study is the imbibition. Let us define what is imbibition. It is the phenomenon of absorption of water or any other liquid by the solid particles of a substance without forming a solution. Now, what do you see in the diagram? Can you see some seeds which are swelled up? So, this is the imbibition process, and the importance of imbibition process in the plants: germination of seeds, developing fruits, and this imbibition process, it depends on water potential, gradient between absorbent and the liquid imbibe. Now, let us have practical demonstration of imbibition. What do you see in the bowl is shrinked raisins. Now, when these raisins are put into water for 24 hours, you can see how the raisins have swelled up. So, this is a clear example of imbibition. So, now let us summarize of the topic plant-water relation. We have studied about osmosis, plasmolysis and imbibition. Have a nice day.